Hello everyone and welcome to IVSA Bidar's official YouTube channel Pashuvani. Pashuvani is a platform that provides you with veterinary education. Our video series consists of practical relevant lecture videos, field approach clinical aspects, guide and tip for future careers, smart content related to both animal and veterinary science. We believe sharing is caring, so please like, comment and subscribe for further updates. And now, let's welcome our speaker of today's episode of Saturday's Sharing.
Hello everyone, I am Somashekar Choudaki, present second year BVSc&AH student of Veterinary College Bidar. It's the time to share about urine and the liver function tests. As we all know, the proverb, health is a wealth. All the living organisms try to maintain their health by different physiological functions. If this homeostasis is altered, then there occurs a problem. Altered homeostasis is result of uni or a multifunction organ dysfunction. The dysfunction is identified by their examinations, and here is a path to know more about urine and the liver function examination. Urine examination. We all know that the kidneys play an important role in regulation of water, acid base and the osmotic balance. The function of the kidneys generally reflected by the quantity and the quality of the urine excreted. Examination of the urine indirectly shows the health of the kidneys. There arises a question why urine examination is important. Means it helps to diagnose or monitor several diseases and the conditions such as the kidney disorders and the urinary tract infections, abbreviated as the UTIs. But also a question of when one has to undergo urine examination. If an animal suffering from such as abdominal pain, frequent or painful urination, sometimes as a part of health urine for examination. For examination of urine, at least 4 ounces of urine is required and well preserved in the refrigerator. If not possible, add drop of toluene or a crystal of thymol is added. The reason for adding toluene or thymol, it merely lies on the surface of urine, forming a thin layer and acting as a physical barrier to air and the bacteria. The urine sample contains cells and the casts sometimes. If those has to preserve means, then one must to add 40% of formalin to 1 ounce of urine. It must be remembered that the casts and the cells will disappear on stagnation. So, fresh specimen is used for examining considered to examine urine. Following are the factors considered to examine a urine. They are color, transparency, odor and the specific gravity. It's an easy demarcation between healthy and unhealthy animal. Urine color is normally transparent yellow in all the domestic animals. It may appear in vivid colors too, like dark yellow, yellowish brown, red, blue and the milky. Presence of urochromes imparts urine its color. The dark yellow color urine is due to deficient intake of water. But there are also reasons for dark yellow color urine like omission, diarrhea and the fever. The pale yellow urine is due to increased water consumption and also in conditions of diabetes mellitus and the insipidus. In case of icterus, the urine takes its color as a yellow, brown or the orange. Red color of the urine is due to presence of hemoglobin and the blood and also in conditions of phenothiazine medication. used for tranquilizer or pre-anesthetic agent. In case of congenital porphyria, urine takes faint pink color. It's a condition occurs due to deficient function of uroporphyrinogen 3 synthase involved in the biosynthetic pathway of the heme. And the greenish color of urine is seen in methylene blue medication. The methylene blue is the drug of choice in case of methemoglobinemia, but it is contradictory in cats and prolonged use causes hench body hemolytic anemias. And the next is the transparency. Transparency also defines health of the urine system. The freshly voided urine is clear from healthy animal when viewed against light in a test tube. The cloudy and smoky urine represents the renal dysfunction. The urine is cloudy if it contains epithelial cells in large numbers and the bacteria and leukocytes as in pyuria and the crystals of calcium carbonate and the ureates. You may wonder that a healthy horse urine is thick and cloudy due to the presence of mucus and the calcium carbonate crystals. The reason for this is mucus prevents calcium carbonate from forming stones. And the next factor considered is the odor. The odor of the urine is mainly due to volatile acids. The excreted urine becomes ammoniacal due to decomposition by the bacteria. The sweetish odor of the urine is due to diabetes mellitus and the odor of acetone observed in ketonuria. A good quality fresh urine of the dog is odorless, but strong ammonia smell suggests occurrence of infection. Compared to all domestic animals, the cat's urine smell so bad. The reason behind this is a fresh cat urine doesn't have a strong odor. However, over time it develops a strong smell. The phenylalanine, an amino acid in cat urine, gets broken down by enzymes into 3-methyl-3-sulfobutane-1-ol, commonly called MMB. This, along with ammonia, produces a pungent smell. It is also to know that a normal urine may form foam if shaken, but the urine in albuminuria condition form plenty of foam. And the next important factor is specific gravity, the term that defines measure of the relative amount of solids to that of the urine. The specific gravity denotes the kidney capacity to concentrate urine by reabsorption. The specific gravity varies inversely to the quantity of the urine. This quick test can help determine how well the animal's kidneys are diluting the urine. The specific gravity is determined by the instrument called the urinometer. The specific gravity may increase or decrease based upon the solids in the urine. In case of cystitis, diabetes mellitus, dehydration, vomition, the specific gravity increases. Whereas in conditions like increased fluid intake, advanced stage of uremia, and the diabetes insipidus, the specific gravity decreases. And here are the normal specific gravity values in different domesticated animals. And the next factor, which includes the chemistry of the urine, is reaction of the urine. The reaction of the urine is mainly depends upon the diet. In vegetarians, the urine reaction is alkaline. The alkaline urine usually contains bicarbonate, carbonic acid buffer excreted when there is an excess of base or alkali in the body. Whereas in the carnivores and omnivores, the urine shows acidic reaction. The urine becomes increasingly acidic as the amount of sodium and excess acid retained by the body increases. Following are the urine reactions shown by different animals. The acidic urine is encountered in following conditions like starvation, fever, acidosis, and prolonged muscular activity. The alkaline urine is encountered in case of cystitis, retention of urine, and rapid absorption of the whenever the kidney's ability to filter the blood by differentiating biomolecules from the waste is damaged, there is a significant amount of them is discharged along with waste in urine. This condition leads to decreased concentration of those biomolecules in the animal body, which is detected by different clinical examinations. Those excreted biomolecules includes proteins, glucose, and the ketone bodies. The first is detection of proteins. The kidneys excrete proteins mainly in the form of albumin. This can be detected by two tests, mainly the Roberts test and the Heller's test. Roberts test. The Roberts test includes detection of protein by reaction between urine and the Roberts reagent. The Roberts reagents consist of 1 part of concentrated nitric acid and the 5 parts of magnesium sulfate. The reaction between the two fluids produces a white ring if albumin is present. Hence, it confirms the presence of proteins. The Heller's test. The Heller's test is commonly used test for detection of proteins in the urine, but it is not so sensitive as the Roberts test. It includes reaction between the 2 ml of concentrated nitric acid and the 2 ml of urine. A white ring at junction of fluids confirms the presence of the albumin. The proteinuria condition occurs in excessive muscular activity and excess ingestion of proteins and in convolutions, which are of physiological causes, and in interstitial nephritis, pyelonephritis, nephrosis, which are of pathological causes. Next includes detection of glucose. Normally, urine does not contain any glucose, but due to dysfunction of the renal tubules, glucose appears in urine. Glucose in urine is called as glycosuria, which can be detected by Benedicts test. Benedicts test. The Benedicts test is the reaction between the Benedict's qualitative reagent and the urine. The Benedict's reagent contains copper sulfate. The glucose being reducing agent converts the cupric ions to cuprous ions, hence the color forms. The reaction proceeds by adding 8 to 10 drops of urine in the reagent and later kept to boil. The presence of glucose turns fluid into green or yellow precipitate. The glucose urea may present in the following conditions: emotional crisis, diabetes mellitus, shock, and hypothyroidism. The next includes detection of ketone bodies. Acetoacetic acid, beta hydroxybutyric acid, and acetone are the compounds that form ketone bodies in urine. It occurs due to impaired fat metabolism, normally detected by Rothera's test. Rothera's test. The Rothera's test includes 5 ml of urine is saturated with ammonium sulfate. To that, add 2 to 3 drops of sodium nitroprusside solution and ammonium hydroxide. A permanganate color is indicative of the ketones. Sometimes, due to damage of blood vessels in the kidneys or in the bladder, produces blood in the urine. Detection of the blood. The presence of blood in the urine is detected by peroxidase test. The procedure of the test is like this. In a test tube, dissolve benzidine and glacial acetic acid. To that, add 2 ml of urine and also add 1 ml of fresh H2O2 and mix it. If blood is present, develops a green or blue color. It is important to know that if blood is found in the last drops of urine, then source is the bladder. If the urine is red throughout, the source of the blood is the kidney. If the first portion of urine is red, then the source is urethral lesions. As like the proteins, due to dysfunction of Bowman's capsule and the fenestrae, the bile pigments also found in the urine. Detection of bile pigments. The detection of bile pigments is normally done with the Malins test. The Malins test includes reaction between 2 ml of concentrated nitric acid. To that, add 2 ml of urine carefully. At the junction, green or violet color formation indicates presence of bile pigments. The liver is an important organ in the animal body, which helps in the fat metabolism, synthesizing different enzymes, and also involved in intoxication. The following facts should be borne in mind while deciding to conduct liver function tests. The first, in diseases of liver, the several functions of the liver are not equally affected, so it may be necessary to do several tests to measure different functions. Second, the liver has great reserve powers, so unless 80% of the liver is damaged, there may not be any evidence of hepatic dysfunction as measured by the tests. The third, it has been stressed again and again that the hepatic tissue has great regenerative capacity, and so hepatic tissue destroyed is quickly replaced. The liver function tests require very good laboratory facilities and all cannot be easily performed under the conditions of the field. The tests that can be easily performed are the iodine tests, estimation of the clotting time, and Den Berg Test. The iodine test. The iodine test requires The reagent, which includes iodine, potassium iodide, and the distilled water, which is kept in an amber colored bottle. The iodine test procedure goes like this. Place a drop of the serum on a glass plate, add a drop of the reagent and stir with a broomstick. In positive cases, floculi occurs at once. Depending upon the intensity of the flocculation, it may be graded as 1 plus, 2 plus or 3 plus. A 1 plus flocculation consists of small floculi. A 3 plus reaction has the whole mixture curdled. This test has been found to indicate the existence of cirrhosis in cattle and the hepatic damage. Estimation of clotting time. Prothrombin is required for clotting of blood, and this prothrombin is manufactured in the liver. In diseased liver, this function is interfered with and so clotting occurs late. So the time taken for clotting of the blood can be used as a test to assess the health of the liver. The test goes on like this. Insert a sterile needle in the vein, taking due precautions. Start a stopwatch when the blood begins to flow out of the needle. Collect the blood about 2 ml on a watch glass. Plunge 6 to 8 capillary tubes into the blood when the blood rises into them by capillary attraction. Keep them on a table and break small pieces of the capillary tube at half-minute intervals. The end point is reached when shred a string of clot is obtained. The normal values for clotting time by this method are following. In cirrhotic cattle, the clotting time was prolonged and was 5 to 17 minutes. Due to some physical, chemical, and biological causes, liver cannot perform its functions. Due to that, there is imbalance in the bile pigment metabolism, mainly bilirubin. The imbalance in the bilirubin is considered a pathologic and a disorder of the pigment metabolism, which results in yellow coloration of the skin and the eyes. The presence of the bilirubin is detected by Den Berg Test. And here is the method to perform Den Berg Test. Make the following solutions. Solution A consisting of sulfonilic acid, concentrated HCl and the distilled water, which is of 1 gram, 15 ml, 1000 ml respectively. And the solution B includes sodium nitrate, distilled water, which is of 0.5 gram and 100 ml respectively. At the time of making the test, mix 10 ml of solution A with 0.3 ml of solution B. This is the Ehrlich's diazo reagent. And next, place 1 ml of non-hemolyzed serum from a fasting animal in a test tube. Run down the sides of the test tube 0.5 ml of the diazo reagent so that it overlays on the serum. In 30 seconds, at the junction of the two solutions, reddish purple ring appears. This is the direct test. If the color develops after 30 seconds, it is indirect test. Mix the contents and add 3 ml of 95% alcohol and mix. If there is pink color now and no color before adding alcohol, it is indirect test. If there was a pink color in the direct test and if it becomes intensified, then it is known as biphasic. The absence of any pink color is a negative test. Please refer the information about differential diagnosis of jaundice in the next slide. Thank you for giving me opportunity to share this knowledge, and thank you for patient listening.